Hi, my name is Megan Leach, and today is 6/21/2021. Sorry. And today we're going to be talking about the regulation of the gastric phase within the digestive system. So, to begin, we need to first talk about what is the gastric phase. So, the gastric phase is initiated before any food has even entered the stomach. This is done through the vagus reflex, which we'll talk about later in detail. And the vagus reflex is from the cephalic phase, which is the phase prior to the gastric phase. Additionally, once food has entered the stomach, stimuli in the gastric lumen will then initiate a series of short reflexes, and this is known as the gastric phase of digestion. So, what goes on in the gastric phase? First, we have a distension of the stomach, and this is because a presence of the food. And then with the presence of food also comes the presence of peptides and amino acids. And this will activate the endocrine cells and enteric neurons to then send on a series of short reflexes. And then we also have hormones, neurotransmitters, and paracrine molecules that will influence the motility and secretion. So the stomach, which is where the gastric phase is going on, has three main functions. We have storage, digestion, and defense. So storage works as holding the food until the small intestine is ready to take it in. This is done through the receptive reflex, and it keeps the upper part of the stomach calm, holding that food, so the bottom part can go through the process and then be ready to go into the small intestine. This occurs commonly when people ingest too much food, which is a very common thing that happens today. The other two functions is digestion and defense. So digestion and defense protects via secretions of the hormones and paracrine molecules. Overall, the goal of the gastric phase is to activate gastric activity and gastric secretion. So throughout the gastric phase, there are 9 different secretions that are secreted into the lumen and act on the food. So those include mucus, bicarbonate, gastric acid, also known as hydrochloric acid, the intrinsic factor, histamine, pepsin, gastric lipase, somatostatin, and gastrin. So I'm just going to give a quick little synopsis of what these 9 different secretions are and what they do within the stomach. So the mucus and bicarbonate are both secreted from mucosal cells. Mucus acts as a protective layer for the endothelium, so it doesn't get harmed by the very acidic part of the stomach. Bicarbonate acts as a buffer for that acid, and it actually, so you have the endothelium, and then you will have the mucosal layer, and then you will have those bicarbonate ions acting on there to neutralize the acid before it touches the endothelium. This actually keeps the stomach and the acid around a pH of 2, while the endothelium cells still remain that pH of 7. The gastric acid is secreted from the parietal cells. So the gastric acid activates the release of pepsin, somatostatin, and additionally it kills any bacteria and denatures proteins and is the main secretion in the stomach that aids in digestion. And then the parietal cells also release the intrinsic factor. When the intrinsic factor creates a complex with vitamin B12, it will permit absorption. Histamine is released from the ECL cells, and this aids in gastric acid secretion. Pepsinogen, also pepsin, is released from the chief cells. So pepsinogen is an inactive form of pepsin. It's released from chief cells. And then once it has been released and active, it will be activated and cleaved to form pepsin. And then pepsin goes on to digest proteins. Somatostatin is released from D cells, and D cells are what inhibit gastric acid secretion, which we'll learn about more later on in the presentation. And then gastrin is released from G cells, and this is what stimulates gastric acid secretion, and we'll see in the next slides how that stimulates the release of the other secretions as well. So how is the stomach stimulated for secretory activity? There's two different ways. As we talked about earlier in the first slide, we have the sight, smell, and taste of food. So this is from the first phase of the digestive system, and this initiates the vagal reflex. The vagal reflex will then innervate with the enteric plexus and then go on to send out postganglionic parasympathetic neurons that will then innervate on target cells for response. The other way is the presence of food. So the food entering from the esophagus into the stomach will cause the distension of the stomach, and then also the presence of the peptides and the amino acids within that food causes sensory input to act on enteric plexuses to deliver short reflexes and cause for the secretion of those different hormones and molecules and paracrine signals. So the result from both of these is the same. It causes secretion and motility within the stomach. So this is an image taken from the textbook, and it looks at how the cephalic and the gastric phase reflexes innervate together to get the same things. So as we see here, right here, we have the stimulation of food. So the stimulation of food causes two different responses. The first one being activation of the vagal reflex. So sight, smell, and taste of food will alert the brain, food, food, food. And this will then go on to activate the medulla oblongata and then send out preganglionic parasympathetic neurons in the vagus nerve to then have a long vagal reflex. This long vagal reflex, as talked about earlier, then innervates with the enteric plexus, and we see here through this gray arrow, then goes and sends out through postganglionic parasympathetic neurons to innervate with target cells and cause that secretion and motility. The second one that causes the same response is when food enters the stomach right here from the esophagus. From there, it then sends out short reflexes that are initiated by the distension from the peptides and from the amino acids. It then sends short reflexes, indicated here by those red lines, to cause sensory input in the gastric mucosa, and then innervates with the enteric plexus. And then as the same as the postganglionic parasympathetic neurons, it will innervate with the target cells and cause secretion and motility. In the next slide, we see how the integration of the two phases can cause secretion. So, as talked about earlier, we see up here, we have a 1 here and a 1 here. These are two of the same phase where we end up getting the same responses. So to begin here, food will enter the stomach and cause an increased amount of amino acids and peptides. This causes a short reflux and innervates with the enteric sensory neuron to then go on and activate G cells. As earlier, we talked about how G cells secrete gastrin. Gastrin from here will go on and activate two different cells. It will activate ECL cells, which release histamine, and also activate parietal cells to secrete that gastric acid. Additionally, the ECL cells, when they secrete histamine, histamine can act directly on parietal cells as well to release gastric acid. From the input from the vagus nerve, we have long reflexes that will go and act on the G cells to then activate, to then secrete gastrin and give the same response as mentioned prior. It also has the ability to send long reflexes and to activate those ECL cells for histamine and the parietal cells for gastric acid. So through both of these, you're able to activate the G cells for release of gastrin, and then go on to eventually secrete gastric acid. Additionally, gastric acid can then go and, through two different ways, activate chief cells and release of pepsin. So through right here and this red line, we see the short reflexes of gastric acid innervating with the enteric sensory neuron to activate those chief cells, release the inactive pepsinogen, which is then cleaved and activated into pepsin. So that's one way. The second way is that it directly acts on pepsinogen, and it will activate and cleave it into pepsin. Pepsin then goes on to break down proteins and cause digestion of proteins. So this in total is how the two different inputs can cause the increase of gastric acid and the release and secretion of all the different hormones and molecules that we talked about earlier. So we need to look at how is this regulated? How is the gastric acid here going to be shut off? And from here we can see in this picture that it is part of a negative feedback pathway. And we're actually going to talk about this in the next slide. So how is gastric phase regulated? There's two main different ways. The first one being the main reason how it is regulated. If there is an excessive amount of acidity in the stomach. So the stomach needs to stay around a pH of 2. If it drops below that, there needs to be a negative feedback loop in order to inhibit the secretion of gastric acid. So the increased amount of acid, as it says here, is the major component of the negative feedback pathway. And what it does, as we look back on this slide, gastric acid can activate D cells. D cells then go on to secrete somatostatin. And as we learned about prior, somatostatin goes on to directly and indirectly inhibit the secretion of gastric acid. So how does it do that? It inhibits it in two different ways. One, directly on the parietal cells to inhibit the secretion of gastric acid. And then it also inhibits the secretion indirectly. And that is through three different cells. The first one is it acts directly on ECL cells, so no more secretion of histamine. It acts on the activation of G cells, so gastrin secretion is decreased, and then it also acts on this activation of chief cells, so the protein digestion will decrease as well. Through this, you're able to decrease the amount of gastric acid being released, and then in return, you can increase the pH within the stomach and bring it back to homeostasis. Another way that the gastric phase can be regulated is through emotional distress. So if there is emotional distress, the sympathetic nervous system would then be activated, and in return, it would override the parasympathetic controls. And this would slow down or stop the secretions going on. So that summarizes the gastric phase and how it is regulated. And I look forward to hearing back from you. Thank you. Have a good night.